Our next stop in the endomembrane system is the Golgi body. This is a stack of membranes resembling a bunch of pancakes. Here's a picture of it here. Now, proteins go from the rough endoplasmic reticulum to the Golgi body when they're going to be made into something special. So what the Golgi body does is it's kind of like a finishing plant. They put the finishing touches on proteins and lipids that are made in the endoplasmic reticulum. The smooth endoplasmic reticulum is where some of the lipids are made. So smooth endoplasmic reticulum makes fats. The Golgi body packages the finished products into vesicles for export. That's the end of our endomembrane system. Our next organelle we're going to talk about is the mitochondria. Here is a picture of one, and here's a scanning electron micrograph showing one, so an actual picture of one. Mitochondria make ATP, which is what the body uses for energy. In the mitochondria, there are many membranes that fold in on themselves. Ions flow across these membranes when ATP is made. This is where respiration, or the breakdown of sugars to release energy, occurs. Mitochondria are not found in prokaryotes. In prokaryotes, they make ATP in the cell walls and in the cytoplasm. The next organelle is kind of a lot like the mitochondria. This is the chloroplast. Chloroplasts are only found in plant cells and a few really weird protists. This is where photosynthesis occurs. This is where plants use sunlight to make sugar. These chloroplasts contain pigments. The pigments are what's used to make to harness the light's energy. These are things like green chlorophyll and also carotenoids, which are the orange pigments in carrots. Lynn Margulis is now a very famous scientist. She is an American biologist of the University of Massachusetts. Back in the 60s, she noticed that mitochondria and chloroplasts were extremely similar to bacteria. And she hypothesized that mitochondria were once bacteria adopted by a larger cell. So they got engulfed by a bigger cell, but they weren't eaten. She hypothesized this in 1966. However, her ideas were not very popular. She tried to publish a paper on this, and it was rejected by 15 journals. A part of it was because she was female in a male profession, but she never gave up. And this hypothesis is now widely accepted, and it's well on its way to becoming a theory. Now that's the inside of the cell. Let's talk a little bit about the outside. Eukaryotes, like plants and fungi, and some protists, have cell walls. These cell walls keep the thing rigid. That's what keeps this false morel from standing up. And what happens is the young plant cell will secrete polysaccharides or sugars around its cell membrane. These sugars then get cellulose extruded onto them. This cellulose and sugar makes the primary cell wall. And some of the sugar is left behind as middle lamella. Don't worry, I want you to be aware of what cell walls are, but you won't be tested on how they form. There's also a secondary cell wall that's made of lignin. This is what's very tough. This is when cells get older and stop growing. They form this thicker cell wall. This is why the young tips of shoots, like on a tree, are nice and green and tender, and they're easily bent. They only have the primary cell walls. The branches of trees, the thicker parts that have been around for a while, have lignin as well, and that's much stiffer. Outside of the cells, there's something called an extracellular matrix. So if you look at these cells, this here is all extracellular matrix. Now this goop is between all cells, not just plant cells, and it's mainly a mix of proteins and polysaccharides or sugars secreted by the cells. Now, cells aren't isolated units. Cells are talking to each other all the time. This is why your hearts beat in rhythm, all the cells contracting as one. This is how you can move your muscles. This is why puberty happens to you all at the same time, all the cells in your body at the same time, that sort of thing. So cells have junctions between them that connect the cells. In plants, there's only one type, and this is because plants have those rigid cell walls. So they actually have what are called plasmodesmata. These are tunnels in between the cell walls of the adjoining plant cells. So the plants can share messages and they can put things back and forth between these channels. Here are these cell junctions in animals. Remember, animal cells do not have cell walls. So these are things like tight junctions, gap junctions, and adhering junctions. Tight junctions are very tight. They seal cells together so fluid can't pass between them. Your stomach is lined with tight junctions. The cells are very closely sewn together. This way you don't have gastric fluid leaking out into your body, which would be very bad. Adhering junctions are the next type of junctions. These anchor cells to each other and to the matrix. So these kind of strengthen the cells and they keep the cells in one place. So this is found strengthening tissues in the heart, so the heart doesn't tear apart when it's beating. This is also found anchoring parts of the intestines to each other. Gap junctions are open channels that connect the cytoplasm of adjoining cells. So gap junctions in animal cells are a lot like plasmodesmata in plant cells. These help send impulses in the heart. The heart uses electric signals, wow, long lecture, to talk to cells. Now the last part we're going to talk about are the cytoskeleton. The cytoskeleton is a network of protein filaments, so like protein threads, that reinforce, organize, and move cell structures. They're made of three different things: microtubules, microfilaments, and intermediate filaments. These are microtubules. They are these big hollow tubes made of little subunits called tubulin. Cute little name. So little protein beads that come together to form a big tube. And these form scaffolding. You can make them and break them very quickly. These are what are used to move chromosomes in cell division. Next we have microfilaments. These are long and thin. They're a thread, not a tube. They're made of the protein actin, so each little bead in it is called actin. And these are used to strengthen or change the shape of cells. So things like amoeba, which I think I have a picture of somewhere. Okay, like amoeba will make a microfilament and then use it to pull along. More on that in the next slide. Then the final one are the intermediate filaments. These are the most stable part of the cell's cytoskeleton. These are like the permanent parts, and they strengthen and maintain the cell and tissue structure. So these things, like the microtubules and the microfilaments, can act like train tracks. And you can get these things called motor proteins. That's this little thing that looks like a pile of spaghetti with an arm. The motor proteins act like train engines to pull stuff, like this big pink thing, along the microfilaments and microtubules. So this is how your body moves things from one part of the cell to the other. The motor proteins burn ATP, so it uses energy. This is an amoeba. The amoeba engulfs its prey by using motor proteins to move actual cytoplasm along the microtubules and microfilaments. So that's how this guy moves. Flagella are another part of the cytoskeleton. These are formed of bundles of microtubules, and they move like a whip. So they're used to move along cells like human sperm. They're long and slender, and they occur singly, so only one of them at a time. Also, there are cilia. These are like flagella, only smaller, and they occur in great numbers. They're on unicellular organisms, like this paramecium here. It's covered with cilia, and it uses it like oars to propel itself through the water. This is a picture of the lining of the human respiratory tract. So this is how, if you inhale some dust, this is in your nose, helps pull the dust back out of your body. And that's it for today.